You wouldn't know it, but we are a lot like ferrets in one particular way.
I was thinking that as I was driving in. Look at all those ferrets I'm driving past. It's unambiguous to me.
Just like us?
Yeah.
Look, there is one particular way that we are a lot like ferrets, and it has to do with this bit of us, this bit between our mouth and our lungs. So in particular, to put it in some sciencey terms.
Not visually, right?
No, not visually, not visually.
I'm going to start staring at you quite a bit right now.
You're looking
no,
for ferretness?
I'm just going to stare at you and see if I can see it.
To put it in sciencey terms, ferrets closely resemble humans in receptor distribution and disease signs for respiratory tract sorts of infections. So this means that their respiratory tract and their lungs behave a lot like ours. So this makes them, as you can probably guess straight away, a pretty good model for virologists to understand what transmission of respiratory disease is like. Yeah, ferrets.
Honestly, I don't think I've ever read or heard of a study that said we tested it on ferrets.
I got a little picture for you of a ferret respiratory tract and a human respiratory tract.
I can't tell the difference. It's true,
and you can't tell the difference.
Hold it up to them so they can...
Yeah, there you go. There's a zoom in. But if you look on the internet, you can see that...
No, but honestly, literally never have I heard, and then we tested it on ferrets. I don't think this is... Okay, I'm interested.
So this is not the story, but obviously someone once discovered that ferrets made a great analogue for our lungs. Anyway, they did.
That's bizarre.
Anyway, in early 2011, two different labs were using ferrets to do some, I'm going to call it interesting things. In particular, they were looking at how flu viruses specific to birds, or, bird flu, could possibly cross over and create pandemics in humans. One of the groups was in...
We've got a lab full of ferrets and chickens right now.
Lab full of chickens, lab full of ferrets to see how it happens.
All of them sneezing.
Yeah, well,
excellent.
Yes, they will be sneezing. So there was one lab at the University of Wisconsin, led by Yoshihiro Kawaoka, and the other by Ron Fouchier at the Erasmus University Medical Center in the Netherlands. They actually published papers that are really quite similar at about the same time. I just want to put both their names in, but I'm going to follow Ron Fouchier's paper first.
We all follow Ron.
Now, I'll read a bit from Fouchier's paper in the Journal of Virology, and I got to do a little bit of sciencey terms here. So just hold
the whiteboard.
Okay, so the title is the multi-basic cleavage site in H5N1, multi-basic cleavage site.
You know how that word gets me going?
Which one, multi-basic?
Multi. As soon as I hear multi, I'm intrigued.
Multi-basic cleavage site in H5N1 virus is critical for systemic spread along the olfactory and hematogenous routes in ferrets. Ignore most of that.
That I can do.
Okay.
Which I understood the conjunctions.
Now, I will do the job here of a science communicator and tell you what you need to know about this, but I'm going to read a little bit from their paper. The highly pathogenic phenotype of avian influenza in chickens is primarily determined by the acquisition of a multi-basic cleavage site, or an MBCS. Don't worry, all they're saying here, if they've got an MBCS, it's a worse flu. And the flu, that's why bird flu spread so much, it had this weird little thing, an MBCS.
And ferrets get it, and we do hang out with ferrets.
Well, no. Here's
ferrets
the thing.
Don't get it.
No, ferrets didn't get it.
You just ruined everything.
Ferrets didn't get it. So birds got this flu. But as Fouchier and his colleagues said, but the route by which H5N1 spreads systematically in mammals is largely unknown. Therefore, we studied the systemic spread of highly pathogenic H5N1 in ferrets, with a special focus on the olfactory route. Additionally, we determined the role of the MBCS of H5N1 in this systematic spread. Now, I'm going to tell you a little bit more about what they did, and it's kind of super sciencey.
Yeah, I'm going to need more details. At the moment, it's a bit thin on the ground.
This is my favorite science paragraph ever, because it's super sciencey, but I'll tell you what it means.
Yeah.
So, highly pathogenic avian influenza virus, A/Indonesia/5/05.
What? A/Indonesia.
They got it from someone. Was isolated from a human case.
Yeah.
They got someone who had bird flu.
Yeah.
And passaged once in embryonated chicken eggs and once in MDCK cells. All 8 gene segments were amplified by reverse transcription PCR, cloned in a modified version of a bidirectional reverse genetics plasmid PHW2000, and subsequently used to generate recombinant wild type H5N1 by reverse genetics.
Seriously, if I had $1 for every time I'd heard a similar sentence, I'd be a multi-billionaire.
I got
PCR.
I got to apologize for that because that was such a sciencey paragraph. But it's something that I read it and I was like, oh, what do they mean here? So I'm going to give you the name that Fuchsia didn't give it. But later on, some people, Fuchsia embraced this name later on. It's called gain of function research. I'll keep going with their methods.
Gain as in
gain.
More of
function. More of function.
All right.
All animal studies were approved by an independent animal ethics committee, and all experiments were performed under animal biosafety level 3+ conditions.
I feel a problem coming up.
Yeah, maybe. Groups of 12 groups of 12 female ferrets that didn't have influenza and were disease-free in other ways were inoculated intranasally in the nose with either of the two.
Female?
Yeah, female.
This is really unusual in animal research. It's one of the main criticisms is they want to check out, how do we know if the pill works? We'll test it on male rats.
Yes.
So the fact they've actually progressed to female ferrets.
Well, female ferrets.
Or is it just because they're going to suffer?
I know nothing about ferrets other than that they're similar to us. But female ferrets, that's what they are. So they gave them 250 micromils in each nostril of either of the H5N1 types. So the one with the MBCS and the one without. Throat and nose swab specimens were collected daily, blah, blah, blah. We euthanized the animals at day 1, day 3, day 5, day 7, alternate animals. And then we did some pathology. So here's the less sciencey version.
Yeah.
Both groups had engineered a novel version of H5N1 influenza. They made a new version of bird flu. And then they tested it on ferrets to see how it would work in mammal lungs. Oh, lungs that are a little bit like human lungs.
Did you say where this was being done?
It was Wisconsin and the Netherlands.
Wisconsin
and the Netherlands. So nowhere near us.
Renowned absolute biological maniacs.
So this small change that they allowed the virus to transmit via droplets in the air made when the ferrets coughed or sneezed. How did they conclude? The multi-basic cleavage site, that little bit of the virus, it previously made a big difference when it was in birds, and they showed it made a big difference when you put it in for the mammals as well. Basically, if you make this tiny change like we did, humans could very easily catch this highly deadly disease. All hell broke loose. Read ahead. Welcome to the Wholesome Show. Science stories for people who sit up the back of the classroom.
Or indeed a little bit too close.
Either way. Very close here.
Very close in computer land.
The Wholesome Show is me, Will Grant.
Oh, and me, Dr. Roderick Griffin. I am not a fucking ferret, Lamberts. I still...
You got to be careful with a sentence like that.
I'm not a fucking ferret.
Just be careful. Don't say it too quickly.
You got to be careful where you put your articles. That's what my mum used to tell me anyway.
The Wholesome Show is brought to you by the Australian National Centre for the Public Awareness
of the Ferret Breathing Tract.
And Science.
Science.
Public awareness of science.
Never heard of it. We
We
need to be more about the public awareness of ourselves. So I'm very worried about where this is going to go.
You're very worried? Are you very worried for my science soul, or are you worried?
Well, I'm also worried that if it's still this full of jargon, I may fall over because there's a lot of jargon.
Don't worry about any more jargon.
I'm going to ask you to periodically reread intro.
Periodically read about the bidirectional reverse genetics plasmid. I don't think I'm going to do that. I don't think I need to. We could use the word genetic engineering there for that, but don't worry about that. Don't worry about that. No, I'm going to tell you a story, and it's a story that asks some really, really, really freaking tough questions for us in the world of science. And it's a story that, look, I think we're going to be grappling with for quite a few years to come.
Are you giving me the impression that it might be relevant today?
It might be relevant today. Even though the stuff that they did in 2011, it might still be relevant today.
Great.
All right.
Well, the stuff we did in 1918 was relevant today, or whenever Trump said the...
1918 was
So why wouldn't that be relevant today?
Nicholas Wade, according to the very brief bio that I could find for him on this website, is a science writer, editor, and author who has worked for Nature, Science, and for many years, the New York Times.
Any of the big ones?
Just the big ones.
He hasn't
I
worked
think.
for Public Understanding of Science. He hasn't worked for the Administrative Sciences Quarterly.
No, but I think if you're going to have the bio, I've worked for Nature, Science, and the New York Times. You don't need to list anything else. I think these are august institutions. Now, sure, he's not immune to controversy.
All right.
7 years ago, he published a book that called A Troublesome Inheritance, Genes, Race, and Human History that was widely denounced by genetics researchers. I haven't read it, but I think there was a bit of controversy.
Genes, race, and
human history.
So when you put the word race in, that normally means it's going to be quite even-handed, non-judgmental.
Yeah, sure.
Yeah.
Sure. I'm not exploring that himself. I'm just saying that he is both august but has some problems.
He has a wide-ranging, inquiring mind is what I'm taking away from this.
Yes, he is.
Yeah, broad thinker.
Anyway, 2 months ago...
Racist, sorry.
2 months ago, Nicholas Wade published an article. So for those of you listening later in time, 5th of May, 2021. He published an essay in the Bulletin of the Atomic Scientists.
Oh, God, that's good. 9/10 of the craziest shit that's called science is in that journal.
It's a great name.
Don't
for
you
a
reckon?
journal.
No, seriously, the Bulletin of the Atomic Scientist. The number of times that's featured in what we've done.
It feels like
I reckon one in every 3 episodes. There's some source, and it's always insane. But still science.
It feels like in the 1950s, some wackadoo atomic scientists said we need a magazine and we're going to publish what. It's not. It's actually really serious. It's a serious newspaper news magazine.
Crazy shit.
I don't know.
Except for this one, obviously.
Okay. He published this essay and it got a whole lot of people to sit up and take notice. So it got me and it got President Biden. So
just the two of you.
just the two of us.
The two most important people in their country.
Probably. Okay. I'm going to read his intro. The COVID-19 pandemic has disrupted the lives the world over for more than a year. Its death toll will soon reach 3 million people. Spoiler alert, that was only 2 months ago, but the death toll will nearly reach 4 million people very soon. I think it's at 3.9 or something like that.
Kicking goals.
Yet, and here's where the rubber hits the road. He didn't write that. I added that. The origin of pandemic remains uncertain. The political agendas of governments and scientists have generated thick clouds of obfuscation, which mainstream science press seems helpless to dispel. In what follows, I'll sort through the available scientific facts, which hold many clues as to what happened, and provide readers with the evidence to make their own judgments. I'll then try and assess the complex issue of blame, which starts with, but extends far beyond the government of China. The virus that caused the pandemic is officially called SARS-CoV-2, but we can call it SARS-2. That's a simpler way of saying it. We're all used to saying COVID-19, but SARS-2. As many people know, there are two main theories about its origin. One,
China. Two, also China. This is going to go that way, isn't it?
No. Theory one is it jumped naturally from bats to people.
Yeah.
Theory two is that it was...
UFOs.
The virus was under study in a lab from which it escaped. Now, it matters a lot which one happened, because if we want to prevent something in the future, then we need to know what happened. Now, before I go into Wade's argument, I just want to pause for something to stress here. I know that this topic has been super, super controversial for the last year for a whole bunch of reasons, and I will go into some of those reasons. And the other thing I want to say is I am not providing answers here, and neither did Wade. And I think anyone who says they have answers here needs to calm down a little bit.
Are you saying just asking questions?
I am just asking questions. I am 100% just asking questions. But these are questions that are not just about big governments, China and the US. They're actually questions that go to the heart of the science that we all do. So, yes, I am just asking questions.
Do you mind? I can make it worse.
No, why?
No, carry on. I'll make it worse though. Carry on.
Okay.
I'm going to make you look bad.
Okay, I'm going to explore the questions and try and ask why some answers might have been provided prematurely and what circumstantial evidence exists for either.
So this is all focusing on the origin. It's all about origin story.
The origin story of SARS-2, of the COVID-19 pandemic.
And this guy has covered it in one article, done.
No, he has asked questions. He's asked questions that I think are important for all of us to ask.
Oh, so he's not asked Fox News questions, he's asked... Am I ruining it? Okay, you tell me. Just tell me the question.
That's part of it. Okay, so let's go through the two big theories and different and how they work. So the natural theory, which has been the dominant one for most of the course of the pandemic, it's pretty simple. it goes from bat to us, maybe with some other animals in the mid.
Goes from bat to pig to food to Gwyneth Paltrow to us, I think is what the documentary originally showed us.
I think Gwyneth Paltrow would count as us.
Big call.
I don't know. Look, we know that there are bat species around the world, but in China in particular, that are super heavy with coronavirus. For some reason, they live really close to each other. They sneeze on each other all the time, and so they are all chock full of coronavirus. We've known that for a long time. So, for example, as I'll come to in a second, SARS-1 and MERS both came from bats originally. So how did this story happen? So after the pandemic first broke out in December 2019, or first was noticed in 2019, Chinese authorities reported that many cases had occurred in the wet market. We've all heard that story.
Yeah.
Just means a market.
It's cleaned often.
Yeah, exactly. It's probably hosed out a lot. So
it is not knee deep in blood all the time.
No, indeed. Yep. But anyway, this reminded people, particularly experts around the world, policymakers, journalists, all of that, reminded people of the previous epidemics. SARS-1 in 2002 and MERS in 2012. So SARS-1 was a bat virus, spread to civets. The animals were sold in wet markets and then got to people. I don't know what a civet is.
It's an animal you wear normally, isn't it? Don't they make gloves out of them or something? They're kind of like minks and things.
Maybe.
I don't know. I'm looking around the room. For those of you listening at home, we have people in the room who are staring at me blankly and or nodding. Okay, so civet you can make a coat out of and apparently eat as well.
Okay, all right, there you go. All right.
I think they're also the ones where you get them to eat coffee and then they shit the coffee out.
I do know that one.
And you get shit coffee beans, which are apparently really good.
Imagine if that was the pathway.
Coffee.
It was the hipsters drinking poo coffee from civets.
Yeah. Have you had it?
I think I have.
So that's a no. You'd know. There's an aroma.
Is it that good?
There's a richness to it. There's a quality.
An earthiness.
There is an earthiness. Cat-like, glove-reminiscent earthiness to it.
Who was I talking? My daughter came back from National Botanic Gardens tour the other day, and she was so excited.
We saw a civet.
No, she was saying because the lady said we could touch the kangaroo poo, and
could or have to?
I don't know.
Dad, I never want to go there again. Carry on.
So SARS, severe acute respiratory syndrome 1, 2002. Yep. MERS, Middle Eastern respiratory syndrome.
Which came from camels, that was confirmed.
Yes, there you go. So the host animal, so it went bat to camel. I'm not sure the pathway of where the bats hit
that's not been fact checked.
Hit the camels, but it did. But anyway, both of those we know came from the same family of bats, and it's the same virus family.
The same family.
Yes.
Literally the same kind of bat. I won't make the obvious gag, but literally the same kind of bat.
Yeah, I don't know.
Is that right?
It's a bat.
Oh, yeah, they're all the same.
Yeah, they're all the same.
Bat racist.
Just stop it. Leave me alone. I didn't look up which kind of bat. It's a cave bat. It's a cave bat.
All right.
Anyway.
I accept your apology.
The virus jumped from... so the assumption here was that SARS-2, COVID-19, jumped from bats, potentially via another animal host, to people. Now, we've all heard a lot of the potential animals here. Was like
Gwyneth Paltrow. We've been through that.
Pangolins. Yep. Or... maybe civets again. I don't know. There were some others.
The pigs.
Okay.
Ferrets.
Possibly. But okay. So circumstantially, we know scientifically that this is absolutely a possibility. We know absolutely, actually, that the virus originally did come from bats. Like we can trace like the genetic origin of the virus.
So we know absolutely possibly?
Absolutely possibly.
Yeah, we definitely probably might know that is...
No, the wild population of coronaviruses come from bats. But at the same time, there is the other possibility. At the same time, Wuhan is also home to the Wuhan Institute of Virology, a leading world centre for research on coronavirus. Now, some people have said... Again, this is circumstantial. It's a little bit like if an outbreak of pizza was to happen just by pure chance in Italy, or an outbreak of croissants was to happen in Paris, or an outbreak of sex offenders was to happen just by chance in Parliament House. It's what those things produce already. That maybe coincidence is a bit of a strange thing to think here. Just one hell of a coincidence. And no, I'm gonna.
Are you talking about all three? Right?
I'm not gonna talk about. I'm gonna drill a little bit more into what supports these theories and what we need to think about.
All right.
So, early on in the pandemic,
starting with Parliament House.
No, not that one. Oh. Early on in the pandemic, the natural theory got two big rhetorical boosts. This is why we all know it as the dominant theory of where the pandemic came from. The first, February 19th, 2020. So this is super early on. I don't think we'd actually declared it a pandemic. We were certainly worried. We were seeing things happening in China.
I feel like we were already abandoning the uni by then, weren't we? That was in March. You don't remember?
It's close. It's close. I don't think we'd gone into lockdown. I think we in Australia went into lockdown.
I know, I did. I left early.
You went early.
I bailed 2 weeks early. It's coming, everyone. I'm out here. I ran with my hair on fire, screaming out.
And I know, listener, for in the future, we're recording amongst lockdown yet again. So it feels a little bit deja vu.
The 8,000 people in the audience right now are all wearing masks and staring at us. It's very unnerving.
February 19, 2020, a group of virologists led by a guy called Peter Daszak. Peter Daszak.
Daszak.
I'm just saying that twice so that you will remember that name.
Do you want to spell it for me? Daszak.
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 He's from a group, probably Daszak, of the Eco Health Alliance of New York. They wrote a letter to the Lancet. The Lancet being one of the big medical journals. It's one of the big ones.
Wade never wrote for that, did he?
I don't know.
Yeah, that wasn't in his bio.
He didn't. Look, the Lancet has done controversies in the past, so they too are not perfect.
Didn't they publish Wakefield?
They did indeed.
The famous MMR autism thing.
Well, this letter might be not similar, but
the same.
It echoes. It echoes gaps in peer review. We are public health scientists who have closely followed the emergence of 2019 novel coronavirus disease and are deeply concerned about its impacts on global health and well-being. That's all fine. We have watched as the scientists, public health professionals, and medical professionals of China in particular have worked diligently, effectively to rapidly identify the pathogen behind this outbreak, put in place significant
I'm going to need
measures.
you to speed up a bit. I'm following it too clearly.
Yeah, he's saying everyone in China is doing a really good job. Again,
public health people agree you guys are tops.
Yeah.
Okay.
Yeah, and they're sharing their results. This is all good.
Yep.
We sign this statement in solidarity with all scientists and health professionals in China who continue to save lives and protect global health during the challenge of COVID-19. We're all in this together. Now, here's the key bit. We stand together to strongly condemn conspiracy theories suggesting that COVID-19 does not have a natural origin.
Proof positive, it does not have a natural origin.
February 19th. It's a conspiracy theory that it doesn't have a natural origin. So he said, scientists from multiple countries have published research and shown research that it came originally from wildlife. No one is doubting that it came originally from wildlife here. That's something important. And also there's letters from august groups like the National Academies of Science and things like that saying that it came from wildlife. Anything else is a conspiracy theory. Now, here's something important. The letter ended with a small little bit at the end. We speak with one voice. To add your support for this statement, sign our letter online. We declare no competing interests. I'm highlighting that so that you can just remember that.
What do I got to remember?
Does Chuck, no competing interests.
No, okay.
The second letter was published...
and his shares in AstraZeneca. Sorry, carry on.
4 weeks later in the journal Nature Medicine. Its authors were...
Another one Wade hasn't written for?
Probably. Nature's close.
It's nothing.
A group of virologists led by Christian Anderson of the Scripps Research Institute. Our analyses clearly show that SARS-CoV-2 is not a laboratory construct or a purposefully manipulated virus. That was...
So all I'm hearing is evidence that it must be a conspiracy because they're keeping it from us.
Look. Firstly, just both of those letters made a big contribution to how the media, the world, us, came to understand the origin of the pandemic. As Wade notes, these two were cited over and over and over again in mainstream press that a consensus of experts had ruled out the lab escape or considered it extremely unlikely. And from that moment on, the natural origin became the dominant story of how the pandemic. So this went through mainstream press and became the understanding that probably most of us have had since that time. I think that's interesting.
Is this where we bring in 97% of climate scientists or something like that as well?
No. Well, no, I'm not going to do that. Okay. They're both flawed. So firstly, Peter Daszak, the Eco Health Alliance, rather than having no conflict of interest, had actually funded coronavirus research at the Wuhan Institute of Virology.
Not even making it difficult.
So
well, it's not like a shelf company with a front and three different Cayman Island bank accounts. Like, oh no, I gave them money. Yeah, that's true.
Yeah, I gave them money, but by giving them money, I didn't mean give them money.
I gave it to them so that I wouldn't be biased.
So I just want to question the Lancet there in their peer review process and allowing something that is a clear conflict of interest, regardless of what's.
But I mean, you and I both done peer review, not for the Lancet, obviously. We do real journals, but the I've never been asked to check the credentials nor the conflicts of interest of authors. Have you?
No, but someone should.
Somebody should do something about that.
Someone should do something about that.
But to be fair, it's not like me, but the idea that everyone should be checking all that.
I get it.
However, it is about Corona, it is about SARS,
it was pretty important. It was pretty important.
Critical.
So second thing, the other Anderson's letter, it made two arguments. One, that they couldn't see a change, and the other, that the virus didn't look very designed. They said it in more sciencey words. Basically, they're saying, well, we can't see any change from the natural population because some older genetic engineering techniques leave a lot of traces. Like you can see it in the genetic code. Unfortunately, there's modern genetic engineering traces that don't leave traces, so it doesn't rule anything out
What's
there.
a trace look like? They're just bits hanging off it.
Yes.
Researchers leave a little tiny signature.
Yeah, it's like those doctors that they have a real brain spasm and they sign their patient.
Oh yeah, they etch their initials into the cortex. Yeah. No, I've seen that.
So actually, not an argument, because there are certainly ways to genetic engineer that do leave it, that can be done without leaving a trace.
All right.
The other one, they said it didn't look very designed because we would have designed it differently. It could be more perfect. You know,
if you bothered, you wouldn't have done it this way. Is not a strong argument.
That's their argument.
Call me skeptical.
So these two both took off, and they became a critical way that we all understood the origin of the pandemic. But it's flawed science. It's flawed science. And I think there's some questions there that we need to ask about peer review, particularly peer review when the science is, well, the arguments are rhetorical and critical in terms of understanding a big emerging threat. Now, doubts started to come about for the natural theory around February 2021. Now, I know that throughout this time there was wackadoos, Donald Trump, other people like that, who were pushing that it was a lab leak or something like that all the way through. I think it's worth acknowledging here that it's kind of, I don't know, it's kind of tricky because Donald Trump and people like that had so many other dumb theories that if they had one theory that was slightly right, it was kind of hard to hear it in all the noise.
It's also the stop clock is right twice a day. You know, you chuck enough shit at the wall, something's going to stick. So
the doubt started to come when the WHO ran a commission to China to say, okay, let's try and look at the origin of the theory. The commission's composition were heavily controlled by the Chinese authorities. Its members, who included Peter Daszak, kept asserting before, during, and after their visit that lab escape was extremely unlikely. So they went there with a little bit of a goal to show that, okay, this isn't what it was.
So they weren't asking if, they were there to prove that it wasn't.
But the problem was, it wasn't quite the propaganda victory that many might have thought. Yeah. They couldn't find any evidence to support the natural emergence theory, which, you know, that's fine. Absence of evidence is not evidence of absence.
They found none.
Couldn't find anything, which is interesting because SARS-1, they found the animal culprit 4 months after the outbreak. MERS took 9 months, and you can imagine how many people would have loved to find the animal pathway, you know, for SARS-2.
So you're probably going to say this, so don't tell me if I ruin the story. Is that because they didn't look well, or there was literally no evidence? Because that
hard to tell. Hard to tell.
Oh, okay.
It could be either of those. I don't believe it could possibly be didn't look well, because I think there are a lot of people outside China, inside China, who would very much like to have found this pathway. It would tell us a lot about the disease, but it would also, as people like Peter Daszak, it would support their argument. So I can't believe.
Did you change his name to Does Suck?
No, I didn't.
I think you did.
Does suck.
Peter does suck is what you said.
I can't believe that they didn't look very hard here. But I think they may have been stymied a little bit in terms of the pathways they could look via the Chinese authorities.
That's what I mean. So you can look hard, but you can't always look everywhere. You can look hard in the areas you can see or are aware of or have access to.
Yep.
Yeah, okay.
So here's the thing. So what was it? 4 months for SARS-1, 9 months for SARS-2, for MERS, and now they were at 15 months. Now we are at 15, 16, 17, 18, something like that, months after the start of the pandemic. And there still remains no easy evidence for the natural pathway to Wuhan.
To Wuhan. Okay.
Yeah,
Okay.
I'll explore that in a second.
Right. Okay.
I'll explore that in a second. But
I thought you meant at all. That's why I was starting to get a bit weirded out. But it's to Wuhan.
Well, yes, we know from Wuhan on, but there's no easy evidence there.
Yeah.
Okay, so how much credence can we give the lab leak theory? Because it has been pushed by wackadoo people. It's been pushed by people that are a little bit insane. So I think it's worth. It has.
Yeah, I agree. I'm not going to argue with that.
What we need to do is understand a little bit about gain of function research. Why would anyone want to create a novel virus capable of causing a pandemic?
I got some ideas. Carry on.
Ever since virologists gained the tools for manipulating a virus's genes, probably from 2011 was a clear marker. People were doing things beforehand, but around then people have gained those tools. They've argued that they can get ahead of a potential pandemic by exploring how close a given animal virus might be to making the jump to humans. Their argument is if we can see what can happen in advance, then we know to prepare for it.
Yeah.
And stuff.
I see no problems with that at all.
And so this justified labs around the world doing this gain of function research. So basically, the idea of gain of function research is you manipulate viruses to make them more likely to infect either humans or other animals, and potentially more dangerous.
But we're doing it, right, to protect us.
Yes.
We're making these super lethal, more dangerous things so that we're going to be okay.
That's the argument. Now, I just want to flag some groups have criticized gain of function research by saying
on what grounds?
Well, aside from the safety, I'll come to the safety in a second.
Okay,
I'll come to the safety. That mutation, there can be so many different mutations. Yes, in your lab, you might have found 5, 10, 15, 20 that might potentially make things worse.
Yeah.
And you've seen what will happen there. Mutations can happen in so many other ways that you can't rule out those pathways. You know, it can happen in literally an infinite number of ways.
It's also the idiocy of suggesting we're just doing the science here, as if there are no other forces wrapped around that may have, let's just say, less than scientific intentions.
Yeah.
Call me cynical.
So what have they done in the past? So they've recreated the 1918 flu virus.
Sweet. Good idea. Yep. Great.
Shown how the almost extinct polio virus can be synthesized from its published DNA sequence.
Fuck me.
Which is cool. You know, you could bring back Tasmanian tigers or bring back polio virus.
Why not do both?
Or woolly mammoths.
Both are traditional. All three are traditional.
They introduced a smallpox. Groups have introduced a smallpox gene into a related virus. So
make it worse.
Yes. So groups have one of the key interests in coronavirus has been looking at the spike proteins, which we've heard many times about the coronavirus spike proteins. So back as far as the year 2000, Dutch researchers genetically engineered the spike protein of a mouse coronavirus so that it would only attack cats, which I think is cool.
I'm okay with that. I'm a dog person. You can do what you want with cats. No offense to cat people.
I don't even know, but it's been happening for a while. So after SARS-1 and MERS, people started studying bat coronavirus in earnest to find out the source and to find out what would actually happen. In particular, researchers wanted to understand what changes needed to occur in a bat virus's spike proteins before it could infect people. Now, I'll introduce you to one of the heroes here of virology, who has done amazing work on the SARS-1 and the MERS epidemics, but may potentially be a villain in this story or an accidental villain. Shi Zhengli, or Bat Lady, as people have called her in
Bat
China.
Lady?
Bat Lady.
Okay.
She is a researcher at the Wuhan Institute of Virology, and she is one of the leading experts in China and globally, actually, on bat coronaviruses. And she has many times mounted expeditions down to the bat caves in Yunnan, which is about 1,500 k's away, to get some bats and get the viruses in them and bring them back and study them. She teamed up with a guy from the University of North Carolina, Ralph.
Batman?
No, Ralph Barrick.
Doesn't have the same
I
ring
know, Batman, Bat Lady. Bat Lady. Batwoman?
Bat Lady's all right. Maybe she's nobility. She could be related to an emperor. Bat Lady and Ralph.
So don't worry about that. The key thing here is teamed up with an American team, which is kind of interesting.
Yeah.
And their work focused on enhancing the ability of bat viruses to attack humans, so as to examine the emergence potential of circulating bat coronaviruses.
Yeah, okay.
In November 2015, they created a novel virus by taking the backbone of SARS-1 and replacing it with one from a bat virus. This manufactured virus was able to infect cells of the human airway.
Sweet, so it worked.
At least when tested in the lab against human cells. So, it kind of worked. They did say, okay, there are risks in doing this kind of research. And they should be weighed against, potential spillovers.
Everything.
So they did say we should be reviewed, and anyone doing this kind of research should absolutely weigh up the risks. It's a complicated thing, this gain of function research.
And it was US-China joint enterprise.
Well, here's the big thing.
On paper. Okay.
So they said we should go carefully.
Yeah.
Because there was a discussion at the time, but the relationship with the US continued because, well, normally we don't necessarily know a lot, and certainly Xi's lab has we don't know a lot since the coronavirus because the Chinese authorities have closed things down. But we do know a bit because they were actually funded by the US National Institute of Health and the National Institute of Allergy and Infectious Diseases, which you might remember is the lab that Anthony Fauci runs. So there's a
What?
I know, right? I know. This going back to conflicts of interest.
It's like suddenly you thought the Joker was dead, but he's back.
No, look,
and Bat Lady's got to fight him. Oh no, she's being funded by him.
Fauci has done amazing work through this pandemic. Absolutely, absolutely. But it's just a little bit weird that potentially Xi's research at the Wuhan Institute of Virology, funded by the US, also required an exemption to be signed off by either the director of NIAID, Anthony Fauci, or the director of the NIH to fund gain-of-function research.
We're going to need a corkboard and some red string now, aren't we? This is
I'm feeling intensely
deep.
corkboard-y, and I'm sorry, because I don't like doing conspiracy theory stuff. What I want to say is...
I think it's excellent.
It's not conspiracy. These are sets of questions that we in science should absolutely ask.
It's not conspiracy if it's true. And so far, all I'm hearing is it must be true.
Oh, I'll tell you later about Martha Mitchell. Do you remember? I'll tell you.
The bread cook. She does sourdoughs and stuff, right?
No, I don't know about that.
I don't know.
Martha Mitchell. Okay, so that's why we actually know a bit about Xi's research, because she had to submit reports regularly to the US National Institute of Health, and it was co-funded by Peter Daszak. So it's all a little bit complicit here. Yeah. Okay, so she set out to create novel coronaviruses with the highest possible infectivity for human cells. Her plan was to take genes that coded for spike proteins and work out which ones would actually work the best in human...
That's fine. I have no notes. So far, it makes perfect sense.
Yeah.
I feel fine.
Now, here's the thing. We don't know. We absolutely don't know. It can't be stated that she did or did not create SARS-2 in her lab, because the records have been seen, but she was
been seen as interesting passive language.
Sealed. Sorry, I slipped. But the thing is, her research points in that direction. Whether or not it happened, her research points in that direction. As Richard Ebright, a molecular biologist from Rutgers University, says, it's clear that the Wuhan Institute of Virology was systematically constructing novel coronaviruses and assessing their ability to infect human cells. It's also clear that depending on the things that they did, the work could have produced SARS-CoV-2. So serious scientists said, look, we don't know because they're sealed, but the work was pointing in that sort of direction.
But this is not, to be fair, unusual work for such labs to do.
No, not at all. No, absolutely.
And that's the thing I think never comes up when you hear these sorts of stories. This is what labs do. It's not that these guys were evil, geniuses working in an underground lair.
Yep.
They were doing what everyone does.
Which is actually a bigger question. It's actually, I think the conspiracy theorists point to us thinking about this as Dr. Evil or something like this.
Yeah.
A weird little lab somewhere that is doing something really, building a doomsday weapon.
Yeah.
Instead, what it might be is labs all around the world might be taking risks that we as a society have not actually talked through enough.
So this is like space travel. The amazing thing is these things aren't everywhere, as opposed to one may have gotten out.
Yes. All
right.
So, yeah, there's an interesting interview that Peter Daszak gave just before the pandemic came out, in which he spoke in, so this is December 9th, 2019. So the pandemic had actually started, but people didn't know about it very much.
Wow.
In which he talked in glowing terms of how the researchers at Wuhan had been reprogramming the spike protein and generating new coronaviruses capable of infecting humanized mice.
Humanized mice.
Yeah. Don't worry about that. Don't worry.
But I want
Don't worry. Humanized mice.
They got little hats.
Just imagine
they can speak. They can.
humanized mice. Just pass it over and imagine. It's just mice that have a little bit of human in them.
Just a science thing. Don't worry about it. Yeah. Just get back to it.
A little bit of
Whatever.
So we can infect them. So we made step 1, make humanized mouse. Step 2, infect it. Step 3,
profits. Okay, carry on then.
And here's the thing, he heard about the epidemic a few days later and then launched into that campaign where he launched straight away saying the idea that this virus escaped from a lab is just pure baloney. It's simply not true.
Baloney. So he used strong language.
He did. Now,
emphatic.
I want to say there are researchers around the world who acknowledge that lab leaks happen, and they happen much more regularly than you would hope. So I've told you stories here before. I told you previously.
They should make them as good as we do hotel quarantine, and then it would be cool. It'd only be 1%.
Stop it. I know. It would only be 1%.
A mere 1%.
A mere 1% of lab accidents. I told you about the Sverdlov anthrax leak, 1979, killed 60 to 500 people. We don't know exactly how many. That's in our Bad Science the Soviets did series. I also told you about Martha Parker, the last person to die of smallpox. That was a lab leak in 1978 in England.
By I told you mean in previous.
Yeah, that was in our Ghost Hospital
That's
series.
That's the Ghost Hospital series.
But I looked at there are stories all over the place of lab leaks.
Yeah.
So
have you done the Roswell one?
No,
you got to get to that. That's been released recently.
Accidental exposure of viable anthrax to personnel at the CDC.
Jesus.
Discovery of 6 vials containing viable smallpox.
In some kids' lunchbox.
No, not quite. At the FDA.
Viable smallpox at the FDA.
I love this one. Accidental shipping of H5N1 from the CDC to another lab. So they sent the wrong virus, and they sent a bird flu virus. In fact, here's the thing: dangerous lab leaks happen nearly every year.
Cool.
And SARS-1, not SARS-2, has leaked. There's at least 6 stories of SARS leaking out of labs.
6.
One from Singapore, one from Taiwan, and 4 times from the Chinese National Institute of Virology in Beijing. So they happen. And in fact, SARS-1 is particularly prone to
leakage.
Escaping. It's a little escape artist.
So it's a property of the bug, not the people.
Well, don't we know? Don't we know that coronavirus, and we're looking at the Delta variant now, highly contagious.
Yeah,
that makes a difference.
It eats through glass.
That makes a difference. If it's a type of it doesn't eat. Well, okay. I'll come to the glass.
You're not going to no.
I'll come to the glass in a second. But a highly contagious virus is more likely to escape from a lab.
Why? If all the protocols
it's in the definition.
But if all the protocols in place, shouldn't it be the same for a lowly contagious and a highly contagious?
Okay. There's two other things that make a big difference. One, if there's a vaccine. If you've got a vaccine for something, then you can vaccinate the lab workers, and protect them from the virus, and then it won't go further. There was no vaccine for SARS-2 for the coronavirus. No, we now have a vaccine, so it could be done. The second thing is, it seems like there's four levels of lab security: BSL 1, 2, 3, 4. 4 is basically spacesuit, and you're doing stuff like.
And you got the external air supply and all that.
External air supply, it's full all the movie sort of stuff.
Yeah,
but it's a pain in the ass. Because it takes you hours to get into your spacesuit.
So don't bother then.
Well, yeah, BSL 3 is a step back, but BSL 2, that just requires sort of lab coat and gloves, and you don't suck up the liquids in your pipette, and you put up some biohazard.
You don't use your mouth. So BSL 1, you suck up the anthrax in your mouth and spit into a test tube.
Yes, that's suck and spit. BSL 1 suck and spit.
I don't think that's a level of safety. I think that should have a different designation.
Don't confirm that. I don't know if BSL 1 is suck and spit, but I'm hoping.
Not your own. They're not freaks.
No, that's when you get the interns to do it. BSL 1 suck and spit with interns. BSL 0.
How does it work? I don't want to work there anymore. Forget the cost credit.
Sadly, people don't not sadly, but people don't really like working in BSL 3, and so they cut corners.
So I'm here to say, I don't give a shit if you don't like it. Get a different job. No, I don't care.
Yeah.
I don't care.
Yeah. Also,
It's like it's like an astronaut saying, "Oh, the helmet, it kind of cuts into my neck, and whenever I get an itchy nose, I can't use it. I'm just going to wear an oxygen mask and goggles when I do something outside the space shuttle." It's stupid.
Yeah, but the astronaut themselves is in danger.
Okay, they die a bit. It's true, but that aside, it's just outrageous. Oh, I don't like it. It's uncomfortable.
It seems that Xi's lab at the Wuhan Institute, they said that because we're doing coronavirus in bats, it can't infect humans, even though they were deliberately trying. So they did most of their research at BSL-2. So no suck and spit, but just wear lab coats and gloves. So they didn't do BSL-3.
Which wouldn't keep the flu away.
Yeah.
Or a cold.
So this is from an interview that she published in Science magazine. The coronavirus research in our laboratory is conducted at BSL-2 or BSL-3 levels. So, yeah.
Is this mimicked around the world? I assume so. Maybe worse.
Let's assume it is. Let's assume scientists are potentially corner cutters when they can. I don't know.
I don't look. I don't care if scientists are people right now. I want them to be like robots. I want them to feel like humans but act like robots because it's
humans at home, robots at work.
That's all I want. That's all. Is that too much to ask?
There's a nice slogan for you. No, it's that. Look, this is a thing that we have to recognize.
That's fucked up, though. That is really fucked up. Like we're dealing with a thing we don't know if it transfers to humans. So let's go low security.
No.
And we're trying to make it go to humans.
Yes, we're trying to make it go.
But we don't know if we've succeeded yet. So we'll just go low security, low safety again. It's perfect logic.
It's easier that way, though.
You've made me disappointed.
I just
probably can't tell.
I just want to let you know that we humans are shitty corner cutters.
God, that's fucked up. They're like, there's no one. Okay, carry on.
Okay, so what do we know?
Well, we know that's. I want to
stress that there is no direct evidence for either of these theories. There is nothing that we know that points directly to the natural origin or the lab leak origin. There is some circumstantial evidence that points in one of the directions a little bit, but it's not enough to actually rule things in or out. But I think the questions that I want to ask at the end still stand. So what do we know? First, the distance. The distance is actually an interesting one because the bat caves down in Yunnan are actually 1,500 kilometers away from the Wuhan lab. And no one has found an intermediary anywhere. So we know that the bats have these coronaviruses that are a lot like that. That's a wild population.
Yeah.
There has not been a human, a canimal, a civet, a monkey, and anything.
A
camel, anywhere between the cave and Wuhan, which is...
correct me if I'm wrong. Bats can fly?
50 kilometers.
Oh, really? And then what? They have to lie down?
They don't go further. Like they're far from home. Like
territorial. Okay.
It's like you got to get back to your cave.
All right. Okay.
They're vampires, obviously.
But they don't do 1,500. They're not like birds. They don't migrate.
It's not even close. It's not like their range is 1498 and one bat. It's 50 kilometers versus 1,500 kilometers. Okay. So there has been no pathway, that animal, human, or anything there. So that's just a question, and I think this is the thing that I can imagine scientists and authorities would really like to find that.
Yeah.
Secondly, there is the evolutionary pathway. In the case of SARS-1, this is the one back in 2002, researchers documented successive changes in the protein as the virus evolved step by step into a dangerous pathogen. After it had gone from bats into civets, there was 6 changes in the spike protein before it became a mild pathogen in people.
Oh, wow.
Then there were 14 further changes. So it was in humans. Then there were 14 further changes before it actually became a virus that was actually dangerous and deadly. Okay. And then it took off. And then there was a further 4. And so we're talking quite a lot of changes. That
fast, right? We're not talking...
I don't know the actual time frame there.
Okay.
But what we're talking about is evolutionary steps that they can trace in a human population. But when you look for the fingerprints of a similar transition in SARS-2, strange surprise because there's hardly any change at all until recently. Once it's been in the population, up until the point when it entered the human population, there's hardly any change at all. So that's one of those questions. From its first appearance, it was well adapted to human cells. Researchers led by Alina Chan at the Broad Institute compared SARS-2 with the late-stage SARS-1, which was by them well adapted to human cells, and found that the 2 viruses were similarly well adapted. By the time SARS-2 was first detected in late 2019, it was already pre-adapted to human transmission to an extent similar to late epidemic SARS-CoV-1. Early strains identified in Wuhan, China, showed limited genetic diversity, which is kind of weird, which suggests that the virus may have been introduced from a single source.
Do you know, call me a crazy person, but I'm starting to see why people might come up with theories.
I
just You just hurt your neck?
No, I'm grimacing with you.
That's hard to not be freaked by if you're not really trying hard to not be freaked.
So here's what I want to get to, because we will probably never know. If we do find the animal pathway, that would answer a lot of questions, and that would be great. We will probably never get any confirmation from the lab. There's 4 different communities that I think have worked against this, and I think it's interesting to pull them apart.
The crazy, the insane, the homicidal, and the suspicious.
No, first you've got the Chinese government. You can guess why they are not interested in showing that they made a mistake that has caused a lot of problems. You can totally understand that. Secondly, the US government is actually complicit. They may have funded this research. They may have gave an exemption to funding gain-of-function research to allow this research.
Which they do themselves anyway.
No, they didn't at the time.
They never did before.
No, they did. I'll tell you in a second. There was a moratorium on gain-of-function research during this period.
Everything's turned to shit. We better stop what we were doing.
Third, some people have argued that the whole community of virologists is maybe a little bit complicit, which is a tough one for us in the science-positive world.
The whole community of virologists?
Well, not the whole community, but those people who are doing gain-of-function research reckon they can make it safe enough.
Yeah, because all you need is you need your lab coat and your goggles and some gloves.
And no suck and spit.
How bad could it be? Carry on.
And then us. I think that there may be a community of people out there who are, us in particular. No, I mean the science positive, the science friendly, that maybe we supported a moment where we were like, oh, it must be natural origin. And we accepted that because A, crazy people were saying the other theory, and B, maybe we accepted leadership from some parts of the science community, the Lancet, things like that, and maybe they were actually complicit. Maybe we enabled this discussion a little bit more.
So the complicit thing, though, it's easy to imply versions of intent with that. Complicit's a strong word.
I don't know.
I mean, it's accidental, deliberate, the other.
So there was a big debate about gain-of-function research. For the research with the ferrets in 2011, the New York Times published an opinion calling it engineered doomsday. And pretty quickly...
What angle did they take?
I don't know. It was a pro.
Balanced piece from...
They were in favour of engineered doomsdays compared with unenginered or non-thoughtful.
Oh, it's true, because if they're engineered, then they can't dooms.
So a group of virologists led by an epidemiologist led by Mark Lipsitch, they put out a statement saying, actually, let's slow down. Let's not do this kind of stuff. Let's make sure we've got a lot of guidelines in place to make sure that we're making the right choices.
What a hysteric.
Yeah, I know. The US government put in a moratorium in 2015, but a group called Scientists for Science, which drives me wild, then said, nah, we can make it safe. Biomedical research on potentially dangerous pathogens can be performed safely and is essential for a comprehensive understanding of microbial disease pathogenesis, prevention, and treatment. A whole bunch of people signed it and said, and I love this, so the leader of this was Paul Dupre. He argued that, sure, there are a few lab leaks, but these are exceptions.
Hey,
exceptions.
Every now and then.
Exactly.
You leak a little deadly virus every now and then.
Sure, it happens most years. Yeah. But that was in the past, and now we don't.
And we're all still here.
Now we don't.
Because we're scientists for science. It's going to be fine.
So here's the thing.
It's not a good argument.
All right. I don't know what the truth of this is, but I do know that we probably stopped the question asking too early in the world of science. And I think that this wider question on gain of function research is actually a much bigger question. How much should we allow this kind of thing? And I read a quote the other day, and this is the quote that I want to end on, that scientists are a highly trusted group of people, but trust can be a double-edged sword. Ooh. There are problems with too much trust. If you trust someone too much, you trust your financial advisor too much, you put all your money with them, they can make mistakes, or they could be nefarious. Is there a danger in which society might trust science too much? Will science be doing the right thing all the time with gain of function research?
I can answer that.
Yeah?
Yes, there is too much, and no, not always.
So there you go.
Problem solved. The other
There
thing
you
I
go.
got to add is I don't know if I care where it came from because in typical crisis fashion, let's just deal with the thing in front of us. First aid says it doesn't matter why the person's lying on the ground shrieking and yelling if there's no visible cause. Deal with the shrieking, yelling, and the broken bones on strange angles.
What if labs around the world are doing the same thing?
No, they would have stopped now.
Well, you know,
just tell.
what if they're doing it with antimicrobial resistance? What if they're doing it with
Spoiler, they are.
doing it with
They are.
algorithms to see, okay, which of these algorithms could manipulate people the most?
I was in such a relaxed mood this morning, and then I came to campus wearing a mask, fogging up my glasses, and now I'm hearing this.
There you go.
You haven't made me happy.
So that is gain of function research, and I think it leaves you with a big question.
Uh-huh.
What should we do in science?
And I was going to say, how long do I stay inside buried under my doona? I'm thinking 3 weeks.
Wholesome Show is me, Will Grant, him, Rod Lamberts, and I'm more worried about the world than I was before.
Yeah,
So
thanks, champ. I really appreciate it. This has been a great day for me.
I'm relaxed now.
That's because I've got